This is a video to help you through the activity on the citric acid cycle, which is also called the Krebs cycle in some textbooks. Here's the overview of metabolism from your textbook. In the most recent activity, we talked about glycolysis and the conversion of pyruvate into acetyl-CoA during aerobic metabolism. There's another figure in your textbook that shows the overall scheme for converting pyruvate into acetyl-CoA or into lactate in the case of anaerobic metabolism. These two diagrams overlap. Glycolysis on this figure is represented by these arrows, and the conversion of pyruvate to acetyl-CoA is represented by this part of the figure here. In this activity, we're going to focus on the citric acid cycle. Notice that pyruvate is converted into acetyl-CoA, and acetyl-CoA goes into the citric acid cycle to produce NADH, FADH2, and ATP. In order for NADH and FADH2 to be converted into ATP, first they have to go through the Electron Transport Chain and Oxidative Phosphorylation. Now let's focus on the citric acid cycle. This looks like a complicated cycle. There are a lot of things going on here. But if you break it down into the simple concepts, it's a little bit easier to follow along. Try to follow along with me now. Here is the structure of acetyl-CoA, where coenzyme A is represented by S-CoA. This part of the molecule is actually this structure. So CoA or S-CoA represents this entire structure, where this S here, the sulfur, is represented by this S. Sometimes an S is written, or sometimes you'll just see CoA. Acetyl-CoA is really just a 2-carbon molecule attached to this larger S-CoA part. The 2-carbon molecule is the part that's important for the citric acid cycle. The last molecule here in the citric acid cycle is a 4-carbon molecule. And this 4-carbon molecule will pick up this 2-carbon piece from acetyl-CoA and it will make citrate. Citrate is citric acid, and this is where the citric acid cycle name comes from, because the first intermediate in the citric acid cycle is citric acid, or citrate. The other product, coenzyme A, can go along and become an acetyl-CoA and repeat this part of the cycle. Citrate is then converted into isocitrate. This is just an isomerization, or it's converting one isomer of the molecule into another. Notice what happens here is that an alcohol group moves from this carbon to this carbon. Isocitrate then takes part in an oxidation reduction reaction with NAD+. NAD+ is reduced to NADH. At the same time, a carbon dioxide molecule is produced, and this 6-carbon piece here loses one of the carbons to become a 5-carbon molecule, alpha-ketoglutarate. I'd like you to take a moment now and notice which other steps here are oxidation reduction reactions. A really easy way to recognize oxidation reduction reactions is by the steps that involve NAD+ and NADH, or FADH2 and FAD. Please recall that we've talked about these reactions before, that reactions that involve FAD or NAD+ are oxidation reduction reactions. There are 4 oxidation reduction reactions in the citric acid cycle, and they are at steps 3, 4, 6, and 8. If you're not sure why these are oxidation reduction reactions, let's take a closer look at step 8. That's this step. NAD+ is being reduced because it's gaining a hydrogen bond. And malate, in this case, is being oxidized because it's both losing hydrogens and gaining bonds between oxygen and carbon. Here's a zoomed-in version of the reaction. Please pay careful attention to this carbon-oxygen bond. The molecule loses 2 hydrogens on malate and gains a carbon-oxygen bond to become oxaloacetate. NAD gains a hydrogen to become NADH, so this molecule is reduced, while malate loses hydrogens and gains bonds to oxygen to become oxaloacetate. So malate is oxidized and NAD+ is reduced. If NAD+ is reduced, it's the oxidizing agent, and if malate is oxidized, it's the reducing agent in this reaction. All right, we just talked about step 3. Alpha-ketoglutarate here is a 5-carbon molecule. In the next step, step 4, we see NAD plus again being reduced. That means that this is another oxidation reduction reaction where NAD plus is reduced as alpha-ketoglutarate is oxidized. At the same time here, we lose another molecule of carbon dioxide. So we go from a 5-carbon intermediate back down to a 4-carbon intermediate. In the first 4 steps of this reaction cycle, we go from 6 to 5 to 4 carbon pieces. Succinyl-CoA has added a coenzyme A molecule in this step 4, and it will now lose that molecule in step 5. In step 5, a molecule of GDP, which is a nucleotide, combines with a phosphate to make GTP. GTP then transfers its phosphate to ADP to make a molecule of ATP. Step 5 here does not directly produce ATP, but it does overall because GTP transfers its phosphate group to ADP to make ATP. Step 5 is a combined hydrolysis and condensation reaction. Let's look at what's happening in step 5 a little bit closer. GDP, which is guanosine diphosphate, combines with a phosphate to get GTP. This is a condensation reaction, but notice that there is no water as one of the products. That's because the water is actually used from this condensation to do a hydrolysis of this bond. Notice that the coenzyme A is not connected anymore to the 4-carbon piece. There has been a condensation reaction happening at the same time as a hydrolysis reaction to form succinate. In the following step, step 6, we produce an FADH2 molecule in going from succinate to fumarate. Once again, this is an oxidation reduction reaction. Step 7 is a hydration reaction where we're adding water across a double bond to break the double bond and make a single bond between these 2 carbons. To do this, you add a hydrogen to one of the carbons of the double bond, and you add an OH group to the other carbon of the double bond. In step 8, we again do an oxidation reduction reaction to take malate into oxaloacetate. Then this 4-carbon molecule goes through the cycle all over again by combining with an acetyl-CoA to make citrate. Take a moment now to answer the questions about the citric acid cycle on your activity. For every molecule of acetyl-CoA that goes through the cycle, you go through the cycle once. And in this cycle, you make 3 molecules of NAD+, one at step 3, one at step 4, and one at step 8. And you also make 1 molecule of FADH2 and 1 molecule of ATP. These are the energy molecules that come from the citric acid cycle. ATP can be used directly for energy, but FADH2 and NADH must be converted into ATP before it can be used as energy for the cell. The conversion of NADH and FADH2 into ATP happens in a process called oxidative phosphorylation, and it happens at a place in the cell called the Electron Transport Chain. This is a cartoon of the Electron Transport Chain from your textbook. The Electron Transport Chain is a series of 5 enzymes that are embedded in the inner membrane of the mitochondria. This is a cartoon of a cell from your textbook. And this is the cartoon of the mitochondria. The mitochondria has 2 membranes, an outer membrane and an inner membrane that is represented by squiggly lines here. There's a space between the inner membrane and the outer membrane called the intermembrane space. This inner membrane is where the electron transport chain exists, and it's represented by this cartoon. Here's the inner membrane, and the phospholipids are represented by these green squiggly lines. This is the inner membrane of the mitochondria. In this diagram, the top space represents the intermembrane space between the inner membrane and the outer membrane. And the bottom of the diagram represents the matrix, or the very core of the mitochondria. Each of these purple cartoons represents one of the enzymes along the electron transport chain. These enzymes act as a wire to transport electrons from one side to the other. And the electrons come from oxidation of NADH and FADH2. Recall that in an oxidation reaction, you remove electrons. At the first and second proteins in the electron transport chain, NADH is oxidized to NAD+, and FADH2 is oxidized to FAD. This produces electrons which travel through the electron transport chain from one end to the other. The electron transport chain is kind of like a wire which takes electrons and transports them across these proteins. The electrons are captured at the other side by converting protons and oxygen into water. This is a very favorable reaction, and it's basically sucking electrons through this electron transport chain. The oxidation of NADH and FADH2 drives the production of water. The flow of electrons from one side to the other side of the electron transport chain creates energy, which is used to pump protons from one side of the membrane to the other. The energy is required to pump the protons because this is active transport. There's a low concentration of hydrogen ions inside the matrix compared to the inner membrane space. The purpose of the first 4 proteins in the electron transport chain are to oxidize NADH and FADH2 and to pump protons across the membrane. This builds a high concentration of protons in the inner membrane space, and they want to come back into the matrix. And that is what the purpose of the last protein is. It's a proton channel which allows protons to flow back down their concentration gradient, and this creates energy. The energy here is captured by converting ADP plus a phosphate group into ATP. The entire purpose of the electron transport chain is to pump protons across the membrane so that they can flow back into the matrix and create ATP. So the reactants here are NADH and FADH2, and they make water and ATP. ATP is now energy for the cell. For every molecule of NADH that goes through the electron transport chain, you can make about 2.5 ATP. And for every molecule of FADH2 that goes into the electron transport chain, you make about 1.5 ATP. Let's use these values to calculate how many ATP is created in the process of glycolysis, when one glucose molecule is converted into 2 pyruvates, 2 ATPs, and 2 NADHs. Only 2 ATP are created directly from glycolysis, but since we're also creating NADH, these can be converted into ATP. If we wanted to calculate the total amount of ATP value here, we would take these 2 ATP and the ATP that could be created from NADH. Since each NADH can make 2.5 ATP, just do a simple calculation to see that there are 7 ATP that come from glycolysis combined with oxidative phosphorylation. This is called oxidative phosphorylation because NADH and FADH2 are oxidized in this process to phosphorylate ADP into ATP. The name oxidative phosphorylation comes from the things that are happening at the electron transport chain. You can use the same concept to do the next calculation. Pyruvate being converted into acetyl-CoA produces 1 NADH. What's the value in ATP of 1 NADH? It's 2.5. Use this information to try to answer the questions on this page of the activity. Check the key to make sure you're correct. Thanks for watching this video on ATP production through the citric acid cycle and oxidative phosphorylation at the electron transport chain. If you have any questions about this video, feel free to post them in the comments. And if this video is helpful for your learning, please let me know by giving me a vote.